So yes, I work at UTS, but I'm also wearing two hats. So from January 2018, I'm a director of the International Research Center at Foshan University in China. So essentially, I'll be talking about a mixed bag of things we're doing in two labs. And also, Mickey gave me extra 15 minutes. I can talk for extra hours, but I don't have slides. So for an exception, I will really probably talk slower than usual, but which is very not typical of me. So I slightly changed the title, but the topic stays the same. We're talking about quinoa as a cash crop for dry and saline lands, dry saline agriculture. And I will also try to make the parallels and to show you, spending a little bit of time, how quinoa and knowledge of the traits quinoa used to deal with these conditions can be used for conventional crops. So hopefully I will be able to convince you. So I'll start with demographic outlook. And what you can see here, it's very interesting because now we say by 2050, we'll have 9.6 billion. This is a prediction. In fact, 20 years ago, or 10 years ago, when looking at these numbers at a token conference, we were talking about 9.3. So we have 300 million more added in the last 10, 15 years to a model, to prediction. And this is because we do not stick to medium fertility rate. It's probably more between medium and high fertility rate. So predictions are, we have probably about 12 billion by end of the century. So they all need to be fed. And the biggest problem which is here is that most of these people are living in countries where the climate is really, really severely unsuitable for agricultural crop production. So this is a very nice paper from Mark Tester and Peter Langridge showing the trends in cereal production, and it was published in 2010 actually. And if we continue business as usual, the blue line goes here, and that is a gap. Again, I mind that it was 10 years ago, and that time we expect less people. So now the gap will become even bigger. So they all need to be fed, and we need to close this gap by doing something about that. Worrying thing is, you see, by the end of this century, Nigeria will be the second after India, and China will be the third one most populated. So some models which I have tells that we'll have 2.1 billion people in India by the end of this century. Agriculture in India relies very heavily on irrigation, and that will be the essence of my talk today. So we need to do something to meet the food demand, and we need to do it under conditions of diminishing resource availability. So we are talking urbanization. This is a Guangzhou, and this is somewhere else, I think more like Pakistan. So you can see that the rural area is shrinking and the urban areas is increasing with time. So I think from the numbers I have, we have now twice less arable land per capita compared with what we had 30 years ago. And it's only going to decrease further because we have a population growth and we don't have much of the land suitable for food production. So we need to increase these food production, step back from business as usual because we'll have a gap getting wider and wider, and we need to do it under conditions of diminishing resources. And it's even more challenging because we have these trends in climate, and we are having agricultural crop production very severely affected by the droughts. I put here a map of China and some statistics for China because it's currently the most populated country, and you can see that the models predict yield losses between 25 and 50% on a regular every 4 or 5 years basis, and this is very serious. It's not really the way. In Australia, which is close to my heart, just the drought a year and a half ago in Queensland, New South Wales, just 2 states out of 7 cost our economy $7 billion. It's very, very significant. So 2008 drought was equivalent to loss of 1%, 1.2% GDP for Australia. And the consequences here will be even worse. So you look what happened under future climate scenarios. And this is a heat map showing the temperature. So what we have here, we have 13 countries which are most affected by salinity. I picked up salinity because I'll be talking about salt stress later. So, but look what happened with temperature in these countries. It's getting hotter compared with median one. And number of drought events, we block them by 20 years. You can see that it's increasing almost like a virus exponentially. And that's a reality of the future. We will have this problem standing with us. It will not disappear itself. And when you look at the models and predictions, we expect by the end of the century 50% of land being dry agriculture. What does it mean? It means we cannot produce anything without irrigation. And that's a reality. And fresh water is also a diminishing resource. Agriculture consumes 70% of all fresh water. And you can see some numbers here. So what if we don't have, or what if it's environmental unsustainable? And that is currently. So there calls, oh, we should stop it, right? We should really just grow plants using rainfall. Impossible. In Australia, a quarter of all GDP comes from only 4.4% of the irrigated lands. So we rely on irrigations very, very heavily, and we need to increase the consumption of water by additional 11%. My Chinese postdocs did some analysis of the FAO stats and other websites, and you can see the trends increase in percentage of irrigated lands in these countries which are required, and you can see the relative withdrawal of fresh water compared with the total water withdrawal. So future agriculture will be impossible without irrigation, and that's a thing we cannot basically skip. We should really plan accordingly. And because of that, agriculture will become saline. Look what happened with the electrical conductivity of the irrigation water. If you look at last 50, 60 years, it's going up and up and up. So there are some standards set up by environmental protection agencies, which tells you how much you can have it. But what is interesting, they have very different standards in different countries. Like in Australia, from the top of my head, we are not allowed to use irrigation more than like 2 decisiemens per meter, something like that. In Israel, I think it's 7, because, I mean, that's a reality. They have no proper high-quality water. So we'll be more relying on this saline water. We'll be more relying on waste waters produced, because fresh water will become diminishing resource and not available for agriculture. So we live on saline planet, so 97.5% of water is saline. And as a result, we put salt into the land with irrigation. That's why quite a few papers call irrigation unsustainable agricultural practice. But I already said that it's unlikely we'll be able to skip it and stop using it, because it's really produce all the food we eat in many countries. So currently we have 11-15 million hectares, and the area is increasing. In fact, as I speak, 3 hectares of the land becomes saline every minute. So during my half-an-hour talk, we'll have another 90 hectares on a planet becoming saline, and not suitable for most of conventional crops which are salt sensitive. And that includes rice, and that includes wheat, and it includes maize, 3 major cereal crops which are responsible for 50% of all calorie intake on a planet. They're all salt sensitive. Barley is better, but we don't really that much barley. So it's already costed us, I think these are data for 2014, 27 billion per year in lost opportunities in agriculture. And again, it will be only going to increase and increase because of these trends I was talking about. So given that we have a shortage of fresh water available for irrigation and the extent of land salinization in the future, we need to make sure that our cultivars will be not suffering from the salt stress and can maintain, well, ideally boost productivity, but at least maintain productivity as we need to deal with this increasing level of salinization in the soil. Again, a little bit of modeling. We have a paper about to be submitted, Trends in Plant Science, a forum paper, and these are some of the results we are presenting them. So this is an expected trend and telling you how many people will be leaving. This is currently where we're standing: China, India, USA, and other countries which are affected by salinity. And this is the size of the cycle, tell you how many people will be living there at a particular time point. Now we have 4.02 billion people, so more than 50% of population living in salinity-affected countries, right, where agriculture is saline. And it's going to be worse and worse, because you see the trends increase in irrigation. Well, nothing to increase in Egypt. It's already 100% on irrigation, but in any other countries, the proportion of irrigated land is going up and up. And when you do some calculations, when you return back to these numbers and look at the EC values, and when you look at the consumption of water added per 1 hectare per year, I took 4 most populated countries here, and you do calculations, you will see that we're adding between 3 and 6 tons of salt per hectare every year with irrigation. And it will not go down because EC only goes up and up and up. So essentially, what does it mean? It means that the land which is arable now will probably become not suitable for traditional crops in 20 or 30 years because we'll be adding more and more salt. And how we selected, how we did the breeding for modern staple cereals, we selected the ones which exclude sodium from uptake. So only 5% of sodium goes into a plant and 95% is pumped back. So as a result, you'll have a buildup of salinity in a rhizosphere, and it comes to such a level that plants will be not able to cope and pump it back again. So that's why I'm here, that's why we're talking about these new crops or domestication or using orphanage species, just to make sure that we move from the traditional breeding, which has no future in 10 or 20 years, to deal with salinity and drought stress. Well, salinity, because drought will result in saline agriculture. So I'm going to talk about physiology a little bit here. And first, I don't really want to sort of say, oh, everything we've done is bad, but this is probably one of the very typical and good examples to illustrate my point. This is a Nature Biotech paper. Oops, sorry. Published by Rana Mans and Adelaide people, Matt Gilliam, Still Time, and all my good friends and colleagues with whom I collaborate and publish papers. So what they did, they followed sort of almost traditional concept of breeding plants. They discovered in one of their ancestry wheat relative, they discovered that the HKT1 gene was responsible for taking sodium from the xylem and putting it back in roots, plays important role in helping plants to survive under these conditions. What they did, they essentially transformed the modern elite wheat varieties and were overexpressed it in a plants, and they went to field studies and they reported beneficial effect, like 25% increase in a tolerance or in a yield, grain yield, under saline conditions. So obviously it's very significant, 25% is good, and this is one of the few studies which went from the petri dish to the field. So it's all great, there's no questions about that. The problem is that when you start to look at the actual data rather than reading the abstract, you see some small issues here. So the positive effect, beneficial effect, was observed only in 1 block out of 3 in a field. So obviously, it does work sometimes, and it does not work in most cases. And most importantly, even if you have 25% increase here, 1.6 compared with 1.3 for control, it's still half of the non-saline control. So we still have 50% yield loss under these conditions. And this salinity was relatively modest. I don't remember from the top of my head here, but it probably was like 8 decisiemens. And the quality of irrigation water right now is probably between 9 and 10 in most of these countries. So it's not good enough. We still lose half of the yield of wheat, even if we overexpress this gene and take sodium from uptake. So this is really, we are back to square one. We need to see what nature did in terms of their creating mechanism for salt tolerance, not what we enforce plants to do. And for 30, 40 years, we were selecting varieties which have no accumulation of sodium in the shoot on assumption that sodium toxicity is the most critical and sodium exclusion is the best way of dealing with that. And I will show you later that this is not how the nature did it. So we don't really have any single species or crop species which we selected, domesticated, which are capable to handle significant enough levels of salinity, which we expect in agricultural lands 20, 30 years from now. So we need to do something about that. And here, quinoa coming into a picture. First, a little bit of introduction. Very healthy from a nutritional point of view. So it's a gluten-free. Obviously, it's very high in some amino acids, like lysine is similar or higher than wheat. It's well balanced. In fact, the FAO call it the most balanced crop on a planet. So I'm not going to go through all these numbers. You can really have a look yourself. But yeah, that's a reality that it's already a cash crop, and I will show you some numbers of production later on. But for me, it's ideal model system to work on salinity tolerance, and I'll explain you in a second why. So why it's cash crop? Look, this is what happened with wheat production in Australia. So we started 150 years ago with very low yield. So at the beginning of the last century, we got like only half a ton per hectare. Later, we had some improvement. Here, we start to apply fertilizers, Green Revolution in the 60s, so we got increase. And here, we start to do something about the agronomy, herbicides, mechanization, blah, 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 mixing with the nitrogen pasture, so crop rotations, and we have increased. But it's still, the maximum we can get now is probably 2 tons per hectare. And when you look at the quinoa, this is really what are reported yields, sort of for some varieties under conditions similar to that. So already now, quinoa can compete with wheat in terms of their quantity per hectare. It has similar or even better nutritional status, and the retail price is pretty significant. This is, I gave this talk in China, this slide. So I look at Alibaba and what you can buy it. So it's between $4 and $10 per kilogram, depending really on the quality of a grain, which is probably higher than, well, not probably, it's higher than the cost of wheat. So it is already a cash crop with a good example. But why I'm talking about that? It's bloody salt tolerant, and it's really tolerant to majority of stresses. So it's ideal plant to be grown under harsh conditions. So it really grows well in different types of soils. You don't need anything special. It can be cultivated in a really marginal soils with extreme pH on both sides, acidic soils, alkaline soils. It's very drought tolerant. I wanted to harvest its seeds from some of varieties and left pots unwatered for 1 month, and they still were green, some of them. So it's really nearly impossible to kill it. So 350, 380 millimeters per year is sufficient for the plant to be grown, which is relatively, well, it's promising. It's frost tolerant. Again, it comes from high altitude in Bolivian altiplano, so let's explain why it has that. And it's facultative halophyte, and it's really salt tolerant. I'll show you next slide why. So this is the picture we took in a glass house when we try to grow the plants with different salinity levels from 0 to seawater. And this is what happened with wheat. So this is a control. This is 150 millimolar concentration of, so this plant will be dead in a couple of weeks and produce no grain. And as you can see here, 150, somewhere between that, it's where the plants is the bushiest and have the highest biomass. So what kills wheat is optimal for quinoa performance. And that really sort of deliver the key message I want to. So we already have the cash crop, which can be used for these marginal salinity affected lands. And this can be a future of agriculture if we put it into the areas which can be only made productive by irrigation. So how and why? And here we're talking about not only the immediate cash return to farmers, but we're also talking about the possibility to understand the mechanisms and introduce them, incorporate them into traditional crops, staple crops we are talking about. So there are many mechanisms, but one of the most important ones is sequestration in these external structures called salt bladders. So these are modified trichomes, just the spherical shape, and they operate as external reservoirs for salt deposition. So they are trichomes anatomically, just modified trichomes, and they're present in theory in every species. So wheat, rice, barley, every plant has trichomes on the leaf surface. The problem is they are not functional enough, or they cannot operate in what they do in quinoa. And I want to comment one thing. Salinity tolerance has been evolved 6 times independently, and salt bladders or secretory glands have evolved 11 or 12 times independently. And that means it's really very critical for plants to deal with salinity load, because otherwise we expect that evolution will go on different things. So we try to put some mechanisms in place and some physiology to explain how it works. And this is a model we predicted with working with Rani Hedrich in TIPS about 5 years ago. So these are transporters we believe are expressed here. My talk was supposed to be after his talk, so I didn't really produce any slides to explain and provide the actual evidence for these transporters, but we did publish a few papers since that time when we validated the operation of these transporters. So how does it work in a lay terms? This is the epidermis of the leaf, and this is a stalk cell. This is very, very interesting. It's a headquarter. It's the cell which controls the trafficking of sodium, potassium, and other ions into mesophyll or into the salt bladder. And this is salt bladder itself. It can be spherical or these funny shapes. So this is again stalk cell in a higher magnification. So the salt goes through this one. This is epidermis. This is a stalk cell. There are a set of transporters here, and this is a bladder cell with a large vacuole. Concentration of sodium in stalk cell can be as high as 1 molar. It's really a very significant amount. And that allows quinoa to accumulate in total about 7 to 10% sometimes of the sodium per dry weight. So it's a very significant consumer of salt from the soil, and that will be my last slide. So potentially it can be used for almost desalination of the soil. But importantly, these transport mechanisms allow plants to get out salt without really any impact or significant impact on their performance, on photosynthesis. So mesophyll is protected, well protected. So we ask the question, well, okay, logically it should be there, so these bladder cells should play the role, but do they really play the role? Because some people might say, look, they're just external epidermis, so they protect against UV or water storage, or they can be something else. So what are the evidence that they're really important, not just, you know, they're curious things? What we did simple brushing experiments when we use very, very fine paintbrush, and we remove all the bladders on every leaf and every stalk and every petiole in a plant, so it was really pretty tedious work. So when we did so, this is the normal leaf and this is after the brushing. So when you did it under control conditions, we don't see any phenotype. The color is slightly different because there is no light reflection when you brushed there. We did metabolomic analysis. Not a single out of 100 metabolites tested was altered by very gentle brushing. So the point is, presence or absence of salt bladders has no impact on the plant performance under normal conditions. However, when you go on to saline conditions, we can clearly see that the brush plants have salt-sensitive phenotype. They're much smaller, they are more dwarf, and they suffer from chlorotic changes and other things. So you can look at the numbers, you can see brushed, non-brushed in terms of the sodium-potassium and relationships. So salt bladders do help plants to survive salinity and calculation, so they take 25 to 40% of their load. In quinoa, it's probably 25. In other halophytes, such as Atriplex or saltbush, it may be up to 50% of the total sodium coming into plant go into these external structures. The second component, which also present in every crop and is underutilized in terms of the breeding programs, is really sequestration inside the plant in a vacuole of the mesophyll cells. So here are 3 components which play a critical role in this process. One of them is really pumping sodium by NHX transporter. So this is a hydrogen sodium exchanger which just pumps sodium in a vacuole. These transporters should be energized, so we have the proton pumps, HATPase or pyrophosphatase pump. And the third component is the channels which prevent sodium back leaking from vacuole to cytosol. So there are many, many papers and attempts by breeders to improve salt tolerance by overexpressing NHX genes. Eduardo Blumwald and sort of Morris Upsey from late 90s, early 2000s, so plenty of reports on brassicas, Arabidopsis, rice, many, many species. It does work sometimes, but not every time. But to the best of my knowledge, there was not a single attempt to target these channels and back leak as a component of the breeding program to improve salt tolerance. And in a next few minutes, I will explain you why it's so important. And that explains sometimes why we still don't have salt-tolerant varieties in the fields, because everyone focuses on this transporter and completely ignore that. Why it's important? Because when sodium comes in vacuole, it can leave vacuole next second, essentially. If there are leak channels, like if you are on a boat somewhere on a lake and you have holes in your boat, you have no time for rowing, you'll be just taking water back and back with a bucket, depending on the size of the leak. So that's happened with plants. They put sodium in, they spend ATP pumping sodium to operate this transporter, and sodium can leave and move immediately. So remember, we want half a molar sodium here, and we need probably 10, 15 millimolar in the cytosol. So gradient is not in favor of us. It just facilitates sodium passive leak. So we need to block these channels, we need to block pathways. Now, what do we know about them? There are two types of channels named slow, or SV, and fast, FV, vacuole channels. Name comes from the kinetics of the response, so when you clamp the voltage in a fast one, the current changes instantaneously. In a slow one, it takes a couple of seconds to get to the maximum of current. They are non-selective, they're permeable to other cations, and difference between slow and fast is that the fast ones conduct only monovalent cations, potassium, cesium, rubidium, lithium, sodium, and these ones also conduct calcium, magnesium, divalent cations. So we ask a question, is anything special about these channels in halophytes and in quinoa so we don't have inner crops? And maybe that's a way to improve, genetically improve their tolerance in plants. And we look at, ask this question, we look at what we know, and we found really disappointingly, no, there's nothing special. It's non-selective selectivity, but 1, so similar amount of sodium and potassium can go through these channels, right? So halophytes do not possess anything unique, right, which crops do not have. At the same time, yet, they are much more tolerant. So why? That means it comes to regulation of these channels, not just something structural. And we look at the regulation, and again, I will skip you the details, so we use patch clamp technique. And here we use quinoa as a model species, and it's beautiful as a model because it has two strategies in one plant. So young leaves have very dense epidermal bladder cells, and therefore they rely almost exclusively on external sequestration of salt. Old leaves do not, so the bladder cells produced and not renewed again. So when the leaf is growing, they stretch apart, so the density becomes less, and later when they're filled with salt, they pop up. And at that stage, plant rely on internal sequestration in a mesophyll vacuoles rather than in external. So when you compare property of channels between this leaf and this leaf, you'll see the difference and role of vacuole sequestration. So we did that, and we found that if plants are grown under saline conditions, they're able to downregulate these leak channels by 3 to 5-fold. So we're talking about biophysical numbers. We're not talking about 20% or 10%. We're talking about 300% or 500%. It's very significant. And we also did some bioinformatic analysis, and what we found, it's very interesting. Again, this is very recent. I think it's only published online maybe like a month ago or something like that. So the slow channel is encoded by the TPCG and TPC1 gene. And when we look at the Podium quinoa, when we look at the Arabidopsis, and we look at a few other species, we found really that there was something special about the structure of the pores, and some loss of amino acid residues in some of these loops in a channel. So we have did not have time enough to explore that, but potentially that's very promising. So there's a hope that we can modify the control of ability to lock these channels in traditional crops as well if we try to identify the functional role of these amino acid residues in a regulatory loop. Why it's important, why I sort of keep telling that it should be locked, why NHX gene of expression is not sufficient. Again, I will skip you the details. We published the New Phytologist Insights very recently this year. So we did some model calculations, and what you really need to do, you need to look at these numbers. If we have 10% of all tonoplast channels open, you have all the energy, 100% of the energy wasted for pumping and it will back leak. When you have only 5% of channels open, we have still 66% of leak. Even when only 1% of channel is open, we have 11% leak. Every cell has about between 200 and 300 channels. So what does it mean? It means that the cell or the plant can afford less than one channel open at the same time, out of 300. Why it's so redundant in terms of functionality, I have no idea. But if we can reduce the channel density by genetic means or find a way to keep them closed, that will really result in a improvement of cost-benefit analysis and possibility to redirect ATP, which is used for futile cycles now for pumping, to something else. So it's very, very promising direction. No one tried it in any breeding program. I'm not sure how to handle it, but at least we are aware about this importance of that. It's not good enough to survive under saline conditions. Plants need to maintain productivity, therefore they need to invest energy, carbon, into newly developed leaves and maintain their normal physiological functions. And when you have this futile cycle, one gene pumps sodium in vacuole and it immediately goes back. You just waste all the ATP available in a cell for this unproductive process. A couple of more slides to show other mechanisms. So we're talking about stomata operation, right, as a critical for CO2 fixation. So under saline conditions, plant will shut down stomata to prevent water loss, and that comes with a yield penalty because they already have reduced CO2 availability. So what happened with plants and how they regulate it? There are two ways. One is developmental, and one is physiological. Developmental means reduction of stomata density. Normally, classical view, when you look at every crop, salinity means reduced growth rate, means smaller cells closer to each other. So when you look at the stomata density, is increasing because the cell's just more compacted on a leaf. And that's what you find in barley. However, when you look at the quinoa and open control, shaded, soil grown, 400 millimolar for 1 month, you can see that leaves, 5 different positions of the leaf, 4 different quinoa varieties, we're talking about 30% decrease in stomata density via developmental mechanism. Later we screened about 120 quinoa accessions and we found the same thing. This plant is capable to control the production of stomata lineage and really developmental processes to optimize water use efficiency. Again, we can learn from it if we understand how it's done. We also have very efficient stomata operation because in fact they do use sodium to change the 2-grain agar cells. So these are direct measurements. We did the epidermal strip assay substituting potassium with sodium, compare quinoa with its glycophytic relatives like spinach, and we did use this electrophysiological MIFE technique to measure the actual fluxes. So you see in epidermis, sodium is not used, flux around 0. In a gut cell, there's sodium uptake, which really drives the trigger pressure in stomata, they open. So the plant managed to avoid toxicity and use sodium as a cheapest moticum to operate the stomata under saline conditions, which crops cannot do it. And the stomata are operating much faster. So here one of my PhD students compared 3 pairs of glycophytes and halophytes, so quinoa, Chenopodium quinoa compared with Chenopodium album. And you can always see that the dotted lines, which are halophytes, are faster than their domesticated, cultivated relatives. So again, we have these differences which also leave us some potential. What was also interesting is it looks like behavior of a stomata is largely uncoupled or independent of the CO2 assimilation in halophytes, and this is particularly because of much higher rate of rubisco regeneration. So it's not really necessarily that you have one-to-one relationship between the CO2 assimilation and change in the stomatal conductance. It's slightly more complicated. So I'll move to the last part. So economic aspect, why it's great, and what happened with quinoa. We see the trend in production, we see a trend in consumption, but look, it's largely South America where it's produced, well, historically, probably for thousands of years. So there's relatively little interest. Again, it's going up every year, but if you look at the total number of production, it's really very, very far from other crops. So this is a comparison with wheat, right? 2015 numbers I got, so we have roughly 0.2 million metric tons of quinoa compared with 700 plus for wheat. So it is not even close, right? It's just several orders of magnitude less. So why? Why is that? Well, there are some answers. You can see, I'm not really sure what percentage on this is, but this is, okay, this is the time when it becomes sort of known to US. Here, when a significant research effort were invested in terms of agronomy things, and here, really when they start sort of commercial production, seriously, and 2013 was in there, it was announced as International Year of Quinoa. So this is where the boost of research and interest become, but it's still very, very far from really being comparable with traditional crops. Several reasons for that, apart from scientific one and lack of marketing. The first one is it has a heat sensitivity, right? So again, it came from high altitudes and mountains. Obviously, many parts around the world, much hotter than that, and the optimal, by the way, I think it's 10 degrees centigrade, optimal temperature for seed germination. So obviously, but having said that, they grow it in Saudi Arabia, so it can be grown in the field, but obviously with yield penalties. So one of the things we need to look at is the genes and mechanisms which will be responsible for improved heat tolerance. Second one is requirement for short day. Again, few varieties I work myself with Sven Eric Jacobsen from this university, and you see there are 4-day neutrals always have short day requirement. This is a killer because some of them just were growing in my glass house, 5 meter tall, hitting the glass house ceiling and still not flowering. So obviously there's a lot of a potential in terms of genetic improvement here, and also there's this limitation because we have combination of stresses. I know that it doesn't like too much hypoxia or waterlogging, and sometimes salinity and waterlogging on irrigation comes next to each other. So there's still possibility for genetic improvement, and I think will be another talk and on that. But what looks promising to me, there is really pretty significant variation. So it is a halophyte, right? So it's salt tolerant, but still we see large genetic variability in this salt tolerance between different accessions. So we screened about 140 of them, and some of them have really dense bladders, some have very few bladders. So we have a potential to really to look at the mechanisms behind the development of salt bladders, stomata, and other things. And in 2017, 3 groups published their genome. I was part of the Chinese group with Zhang Kanzhu in Shanghai. Another one was KAUST, and was the third one. So we do have genetic tools now to do it. We have all the information we need, and we have pretty significant physiological basis, so we really understand what we are after. So my techniques which I apply allow us to do cell-based phenotyping, so we can really measure the ion transport from this single stalk cell or bladder cell, and we can really understand which genes are behind that. So my last slide will be about sustainability and why I'm putting that as important things. As the way we did the breeding, as I said, it was for sodium exclusion, and as a result we have the progressive build-up of salt in a soil in a rhizosphere. If we continue to do as that, so currently we're standing at about 10 tons of salt per hectare in a normal soil, right? Most of them is just bound to the cations in the clay, so obviously it's not really making any harm. If you continue for 30 years putting that irrigation water I was telling you with that DC levels, and you do no exclusion, you end up with 10 times higher salinity level in the soil, which will be completely unproductive. So in 20 years, we'll run out of production on that paddock. If we use wheat here in the red, it helps very little because it does not accumulate salt in a shoot. If we go for quinoa, we'll end up about here, but there are other halophyte species like Atriplex, 2 Atriplex species in these colors, which can almost reduce accumulation or buildup of salt or even cause decline because they accumulate more than we put with irrigation water. So the point I want to make is we need to breed our staple crops for sodium inclusion, for halophytism, to get the sodium accumulated safely, deposited safely. So we do kill 2 birds with the same stone. We have still maintained high production and at the same time prevent land salinization. So we do responsible, sustainable agriculture. The trends in breeding we did follow last 30, 40 years in salinity, from my point of view, are wrong. They are short-term solution. They allowed us to maintain productivity. Right now, they'll probably be good enough for the next 10, 15 years, and after that, we'll come to a point when plants cannot cope with anymore. So I think that's a good time for us to rethink our concept and do the paradigm shift in how we select varieties. We do not have select them for low sodium. We need to select them for high sodium accumulation and at the same time high yield. And this is non-orthodox approach, but I think we are getting more and more in agreement this is a way to go forward. I think I'll stop at that. I'll nearly use my time again. Thank you.
Thank you very much. So, questions for Rosa Lopez?
Yeah, thank you for a fantastic talk. I was actually thinking, because you had this little table on the different varieties which are short day and which are kind of daily neutral, and of course, I mean, all the Danish varieties seem to be day neutral. Do you think if there's any kind of penalty? Because it's obviously something that we have bred somehow in the adaptation to the Northern Hemisphere somehow.
Yes, will be. I don't think we did really single factorial experiment properly to answer that question. I know for sure that actually I was a co-supervisor of PhD student from this university on that project. And it's a carbon, it's a sugar which serves as a signal. So obviously we are talking about very interesting things, sugar as a signal, sugar as energy source, and it comes to yield penalties. But indeed these varieties which are day neutral, they are a result of produced at this university or result of selective breeding, they are their phenology is very short. I mean, they produce grains after 2 months only. So they are typically smaller, and therefore their productivity is not as great as when you have 4 or 5 months of developed their bigger leaf area. To what extent it's controlled by the day length, I have no idea, to be honest. But that's, for me, that's heat tolerance and this day length of photoperiodic requirement are two of the most critical things. I'm not talking about agronomical trials like pre-harvest sprouting and other things. I'm talking about physiological things. But this is really what limits its distribution, let's say, in Australia. Some parts of it is very short, and the day is too long to be put in a field for the grain production. Having said, some farmers come to my lab and say, oh, we are happy to take it because we have part of a field which is saline and nothing can be grown there. So we can grow them, the silage to feed animals, like dual purpose use.
Okay, I have a question first. When you showed the effect of brushing, the plants were very small. I mean, before and after brushing, you had the salt tolerance. I mean, normally quinoa plants can grow quite big, yeah? So I just wonder, is it an effect you only see in small young plants, or is it also in older plants?
I mean, it just explained the pot was 6-inch diameter, so just a limitation on the root volume, and therefore that's how they grow. There's no really, I mean, we did many different experiments. If you do severe brushing, damaging the leaf, so we have mechanosensory channels activated, you have the stunting of the growth, you have a phenotype. But we did it very gently, so if you do that, you have no difference between the brushed and unbrushed controls. So the size of a plant is determined by the time of the year they were planted, because they really start flowering early, and also very, very small size of a pot. So we have restriction in root volume. It's almost like a bonsai effect related to cytokinin production in roots. Yeah.
So and then I have a question from the virtual audience here. So how can we best compare the Pokali rice and the quinoa salinity mechanisms? You know the Pokali rice?
Yes, I know Pokali rice. We work with rice as well. Pokali is, I would say, if you put 80 millimolar sodium chloride, you probably have about 60% of yield penalty. If you put 100 millimolar sodium chloride, Pokali most likely will produce no grain or maybe 20%, and quinoa has a maximum production of that. So rice is one of the most sensitive species to salinity. 10 millimolar sodium chloride already caused some yield penalties. So they're not comparable, simply not comparable when they're grown and the salinity is high enough to call saline soil, which is 4 decisiemens per meter of 40 millimolar sodium chloride as a threshold. So most of the rice, including Pokali, will have, well, Pokali is one of the most tolerant genotypes, so probably it will have 70% of control yield, but still 30% reduction.
Okay, thank you. So I have another question from the audience here. It's from Gabriela Alandia. Have you studied any relationship between saponins and salt bladders? I mean, because usually salinity-tolerant materials have high saponin content.
Sven Erik did that, so he had a student here at the University of Copenhagen working on saponins. I was not a part of that project, but I can tell you one thing. When you look at this genetic diversity of quinoa species, and we have now about 320 accessions in our collection, some of them really red, really dark red, some of them pink, and some of them white. So I expect that the amount of anthocyanins or betalines, because that's what they cause a color, will have play some role in responses to salinity. as potential non-enzymatic antioxidant or regulator. And I would expect that it will have correlation with the saponins as well.
Okay.
But I haven't really done anything on the quality of grain.
And a new question from the audience here, so from Klaus Krog-Madsen. So would it be possible to separate the saline trichomes and return remaining biomass to the soil?
Yes, it would be possible, processing it. So the idea of using halophytes as a forage species is old, maybe 40, 50-year-old. The problem is that most of the halophytes in their leaves, they accumulate very significant amount of oxalates, which really ruins animals' kidney rapidly, 2 or 3 weeks or months, and basically so you need to mix it. But the possibility of improving processing mechanisms is one of the things. And with quinoa, most of the salt go to bladders. You can mechanically remove it in, blender type of things. They really stay amazingly robust and alive. You brush them and you cannot really crush them. They have enormously thick cell walls, so it's very difficult to work with them in terms of the impalement. I think we almost failed to do the pressure probe measurements to look at the Tugger relations. We cannot impale them, but that gives advantage. You can really separate the biomass with no salt and the salt load and sort of separate them into compartments and return back organic matter to a soil and remove that small part which can accumulate soil, put in sea if you want, back to where saline belongs. I think it's possible.
Thank you. So, Pablo?
With respect to the effect of temperature on germination, so the problem with temperature is only with germination or in other parts of the developmental program as an adult plant? Or
That's a good question. What I showed, I found that on one of these websites, company which produce quinoa commercially, and they say that optimal temperature for germination 10 to 12 degrees centigrade. I'm pretty certain that the temperature will have impact on other parameters, like phenology, developmental things, but I'm really stress biologist and I'm developmental biologist, so I cannot really tell from my experience. I would probably say by common sense, yes, it will have impact on other things. I would say at the low temperatures, probably the lifespan will be longer, which probably allow to produce high leaf area, and therefore, assuming the same harvest index, you'll have more grain. And that's one of the problems in Australia, because as I said, sometimes Mickey's question was why they're so small. Well, because some of them start to produce seeds 1 month after, or flowering after 1 month after planting. So the plant is still small, and already there we have too warm climate. Maybe day length, maybe combination of day length and temperature. So it's obviously a very interesting issue to follow.
Okay, and do you think dormancy, those seeds that are more sensitive to temperature are acquiring dormancy, and that's why the germination is lower?
I can tell you that there's almost 0.9 correlation between the dormancy and salt tolerance. The most tolerant varieties which we have, so as I said, we have 300 accessions we purchased from a gene bank, and we phenotyped 140 of them. In maybe 5 or 6, we could not really get germination. It's just seed dormant, and they are the most salt tolerant. So obviously there is something in there. I don't know, maybe ABA, because it's a stress hormone, and high ABA content is sort of what behind a dormancy. So we try to do the GA treatment of seeds that does help a little bit in terms of germination, but still, again, it was sort of on periphery of my work, but I'm thinking about that, because usually we have a trade-off between the tolerance and productivity, and we need really to overcome that.
So, Fernando Geo Flores?
Sergey, I was wondering whether the plants that grow in the Bolivian and Peruvian altiplano are subjected naturally to salt stress. So
yes.
Why do they evolve, and where does the stress come from?
Well, there are some, again, I haven't been myself personally in that part of the world, but Sven Erik spent probably 10 years collecting these seeds around. So there are some high altitude lakes with pretty saline soils around them. So quinoa is growing there. And also, these soils are very impoverished in terms of nutrient status. There's almost no nitrogen, no phosphorus in there, and also high UV radiation. So the salt tolerance has three major components. One is osmotic adjustment to deal with water stress deficit, another one, iron toxicity, which quinoa resolved by pumping it into these epidermal external structures. And the third one is oxidative stress, because ROSs were important developmental signals, but they produce unbalanced, they activate channels. So more sodium coming because there are ROS-inducible uptake channels, non-selective channels, and potassium leaking out. So being grown at 3.5 kilometer altitude with a lot of UV, quinoa developed very efficient ways of dealing with ROSs, and that explains salt tolerance. So it's not necessarily soil salinity in the soil, it's indirect YRS homeostatic control, which make it beneficial on the ground level.
So I have a question. So you said they like salts.
It's benefit from, yeah.
But if you grow them on non-saline conditions, yeah?
They still grow, yeah.
What cultivars would be most productive, the salt tolerant or the non-salt tolerant?
Salt tolerant, no, I think non-salt tolerant. Well, sorry, Mika. They are all salt tolerant, right? It's a halophyte. So the photos I showed, it's these Danish cultivars, which are one of the most salt sensitive. They still benefit from 100 millimolar sodium chloride, right? Some extreme halophytes benefit from seawater. So for Sarcoconia, perennial halophyte species, 400 millimolar, 80% of seawater is a maximum for productivity. So these ones are still halophytes. They benefit from salt, but they benefit to different extent, and it's a facultative halophyte. That means it can be grown without the salt, but the yield will be less than in the presence of some salt.
Okay. Thank you very much.